Okay, today we're going to start with our worksheet which is about ChIP-seq. So we did go over this briefly last time, but I think it was kind of fast, and another example certainly won't hurt. And then we want to talk a little bit about Xist and how it's believed to spread along the length of the X chromosome. Okay, so this is our last worksheet, yay, and it's about a ChIP-seq experiment, and involves proteins that are expected to bind different regions of, it says human, but it's actually yeast DNA. So in this case, we're going to be doing different antibodies. The previous example, we just had antibodies to acetylated histones, or methylated histones rather, but this time we're going to include an antibody which is directed against RNA polymerase. It's also a little more complicated because we're going to involve modified lysines in yeast with wild type versus mutant strains. So it's a little bit more difficult to look at the original picture, but we'll step through it. Okay, so here's the result of our ChIP experiment, and it is sort of overwhelming to look at first, but you just have to break it down into its component parts. Okay, first of all, we're going to be looking specifically at genes at a particular region of chromosome 12, and the coordinates are given above. The actual gene names are given on the x-axis. So notice we've got two different experiments going here. In one experiment, we're going to look at the effects of mutation of an acetylase called ISA1 on acetylation of histone H4 in its vicinity, and also its effects of that same mutation on RNA polymerase II occupancy of those genomic regions. In the second half of the experiment, we're going to do the same thing, only we're going to look at whether or not GCN5 obliterates lysine 9 of his3. So we're going to this involves a different histone, it's H3, and a different acetylase. But we're going to do the same experiment, looking for the absence of acetylation in that region in the mutant strain, and also looking at RNA polymerase II occupancy. Okay, so the ChIP part refers to chromatin immune precipitation. The seq part refers to the fact that we're going to sequence the precipitated DNA molecules by sequencing. Okay, so remember that we're going to be immune precipitating using proteins that are bound to the DNA. So the DNA is actually just coming along for the ride at this point. It's not being directly immune precipitated by itself, but ultimately it's a sequence of that DNA that's going to tell us the genome locations where the acetylation was happening and where RNA polymerase was binding. So we're going to compare the acetylation and RNA2 poly2 occupancy in the genome of wild-type yeast compared to mutants in these two acetylases, GCN5 and ISA1. Okay. So remember that the plus means that the yeast cells are wild-type normal for these genes, and the minus refers to the fact that the cells are mutant and that they lack these enzymes. So we want to know what's going on with these genes in terms of polymerase transcription and occupancy along the x-axis at the different genes. Okay, so there's a lot of steps to this. This is going to be yeast, so we're going to grow our cultures, obtain cultures of the yeast. We need 3 genotypes. We need wild type for both, mutant for GCN5, mutant for ISA1. So already we're going to have 3 different samples that we're going to have to be dealing with. Then we cross-link to the proteins to the DNA using formaldehyde, and this will of course cross-link any proteins that are present, not just the ones that we're going to be immune precipitating. Then we break open the cells and shred the DNA in one way or another to form smallish pieces, 200 to 500 or so base pairs. Okay, so now we're going to split our extracts and add different antibodies according to what we're going to be looking for. Okay, in this case, we need antibodies that are specific for the acetylated forms of H4 and the acetylated at lys 9 position on H3. We also need antibodies specific for RNA pol2, presumably one of the subunits of RNA pol2. Okay, so now after we've gotten our antibody, we're going to separate our broken protein plus DNA mixtures according to which antibody will be added. So we need to have a separate extract or a separate sample for the wild type versus the mutants, and also for the different antibodies that are going to be added. So these are going to be added separately. So when the antibodies are getting into contact with the proteins in your extracts, then they will bind to them, and we need to separate those complexes from the rest of the chromatin and the rest of the DNA. All right, so one trick that is very popular is to use proteins A or G that are bound to inert beads, usually either sephrose or metal. Okay, so these proteins will bind to the antibodies to form a complex in which you will have your protein A or G hooked to the bead, which can be centrifuged out because they're big, and those are bound to the antibodies, which are bound to the histones or the RNA polymerase, which in turn are bound to the DNA. So the DNA will come along for the ride after the binding occurs, and we centrifuge them out to pellet the complexes. So the complexes you get include the DNA, bound to the target protein, bound to the protein AG, bound to the beads. Okay, so now we wash really well to make sure we get rid of any extra DNA proteins that are present. And then we release the DNA from the proteins by reversing the crosslinks. So now the DNA is ready for sequencing by next-generation sequencing methods. Okay, so what is produced from this is a gigantic number of short sequences, most of which will occur at unique locations in the genome. So these reads, or sequences, are aligned to the known genome sequence using very complicated software. So even a very short read can be uniquely aligned to a specific location in the genome. And that's what the software is going to do for us, and then display it in a user-friendly way. Okay, so the software displays the results of the sequencing reads as peaks. So imagine it makes a little tick mark whenever it sees a read at that particular location, and it gets more and more tick marks. So higher peaks mean that there were more reads at those locations, right? So this actual experiment would give us reads across the genome, not just the region we're looking at. But the region we're looking at is represented as coordinates on a particular chromosome, and we're looking at a group of about 9 genes on chromosome 12. Here's what the results of our ChIP-seq look like. So we can take this apart and look at different aspects of it in order to answer some questions about how this all works. So we can see in the ESA1 wild type versus mutant experiment, we can see we're looking for histone acetylation on H4. And we can see in the wild type strain, there are peaks that are strewn across that whole region. There's some regions that don't have very much, but there's some regions that really have quite a lot. But in the ESA1 mutant, those peaks have disappeared. So that tells us that in this region of the chromosome, the ESA1 mutant is unable to acetylate histone H4, which we expected. But now we look at polymerase II occupancy. We can see in the wild type strain, we can see that there are peaks of RNA polymerase, again, all across the region, with different parts of it having a lot more RNA polymerase than others, implying that those genes are less highly transcribed than others. But we look at the effects of the ESA1, and we can see the same RNA polymerase peaks in the same regions in the wild type versus the mutant. So that tells us that the ESA1 is not required for acetylation, for RNA polymerase occupancy along that region, even though it is required for H4 acetylation. Okay, the GCN5 experiment is basically parallel to that, only we're looking for acetylation of lys9 on histone H3. So in the wild-type strain, we can see in the wild-type strain for GCN5, and we do our pull-down, we can see we get peaks all the way across the region, implying that those histones are acetylated because they came down with that antibody. But if we look at the mutant strain, there's very little or no histone H3 lys9 acetylation across that region. There may be some. So that tells us that GCN5 will acetylate across that region, acetylating those histones. But if we look at RNA polymerase occupancy, again, we see that even in the GCN5, in the absence of that histone H3 lys9 acetylation, RNA polymerase still occupies pretty much the same regions as it did in the wild type. Okay, so now we can look at our data and answer a few questions. Are the histone H4 proteins in this chromosome region acetylated in the wild-type strain? So we can look at the peaks and say, yes, there are peaks across that region, so they are in fact acetylated. Are the histone H4 proteins acetylated in the ESA1 mutant strain? Well, no, because there's hardly any signal at all, so we would say no, they're not acetylated. Is ESA1 necessary for RNA pol2 to bind to the region? Well, no, because the RNA pol2 peaks are very similar in the wild type versus the ESA1 mutant. So RNA polymerase is able to bind perfectly well in the absence of the acetylation that's promoted by ESA1. So is histone H3 acetylated at lysine in the wild type? Yes, we see peaks all the way across the region, indicating that those histone H3s were acetylated. Is histone H3 acetylated at lysine in the GCN5 mutant strain? Well, very, very little acetylation is seen, so no. Is histone H3 acetylation necessary for RNA Pol2 binding in the region? The answer is no, because RNA Pol2 seems to occupy pretty much the same regions as it did in the wild type, even if the GCN5 protein is not present. Okay, so in general, are the histone acetylation levels correlated with RNA Pol2 occupancy in the various genes? Please give an example. Okay, so to answer this, you just look at regions where you have high acetylation and look to see whether those same regions have high RNA Pol occupancy. And conversely, if you look for a region that has very little acetylation, it will likely have very little Pol2 occupancy. So this doesn't require too much thought, just do a few comparisons. And the last question is, if acetylation of histones is so important for gene activation and transcription, how is it possible that the loss of acetylation seen in the mutants has little or no effect on RNA Pol2 occupancy? Okay, so there are several reasonable explanations for this. Okay, so the answer really is, if you think about the histone tails, there are numerous sites for acetylation. So knocking out one particular location of acetylation doesn't mean that those histone tails aren't actually acetylated. These are redundant in the sense that you can acetylate numerous locations, and it may well be that some sites are more important than others, and in any case that some sites are not important in the sense of activation of transcription. So just make up some reasonable explanation for that. Okay, so our real topic today is Xist coding of the X chromosome, and people are still figuring out really how this works. Okay, so we're going to consider two ideas about how the Xist RNA that starts at the X activation center manages to make its way all the way across the whole chromosome. So two models are under consideration, really. One is that it starts at the XIC and then spreads out in a uniform way, diffuses basically along the length of the chromosome. Well, it turns out it doesn't do that. Rather, the distance between Xist and the particular site of the Xist gene and the site of the RNA, it doesn't matter too much, because sites that are very far away get coded at a rate that's very similar to the close ones. So the question is, how could that possibly work? Okay, well, the answer is it has to do with the three-dimensional organization of chromosomes into various looped domains, in which we'll discuss this next time. But the idea is, if sequences are far away in a linear chromosome, in three-dimensional space, they can be quite near each other. So that's the idea that's into consideration. Okay, so the model 1 has it that it all starts at Xist, and therefore would spread uniformly along the length of the chromosome, starting at Xist, where distant sites would get occupied much later than closer ones. The other model, which seems to be close to correct, is that Xist starts, of course, at its own chromosomal DNA locus, and then basically hops to different regions along, some of which are quite far from Xist, in very short amounts of time. Okay, so the way this is believed to work is that those pieces of DNA that are far on the chromosome can actually be brought to proximity of the Xist DNA, and therefore the RNA, and in that way basically have the Xist delivered to the different locations over fairly large distances over a fairly short amount of time. Okay, so in the top model, it's diffusion, or else spreading goes to distant sites, filling in after that. Okay, so the answer does seem to be spreading goes to distant sites early on, based on physical proximity because of the chromatin structure and chromatin looping. Okay, so how to watch Xist spread along the X chromosome is going to involve another high-tech ChIP-like acronym called RAP, RNA antisense purification. Okay, in this case, it's the similar idea. We're going to have cells which are expressing the Xist. It's going to be an inducible system, as you'll see in a second. But the probes, instead of being antibodies, are single-stranded DNA, which are complementary to different parts of Xist. Okay, so we need to label our probes so that we can pull them down. And these are going to be labeled with biotin for the purpose of precipitation with streptavidin-coated beads. Right, so the idea is to capture the DNA to which the Xist RNA is bound, and then sequence it to see where Xist is found along the X chromosomes at different times. So this is similar to a ChIP experiment, but with the twist that you're going to be using labeled DNA probes that are going to be pulled down by the streptavidin biotin trick. Okay, so the streptavidin biotin trick is widely used. Streptavidin is a protein that binds extremely tightly to biotin, which is a small molecule. And this can be used for precipitation purposes because the interaction is so strong. So in this case, we can use biotin-labeled nucleic acids, or it could be biotin-labeled anything really, and proteins will bind very tightly to streptavidin-coated beads, allowing them to be purified. So it's like your basic IP type experiment, only instead of using an antibody and protein A or something, you're going to use biotin-labeled nucleic acids and use streptavidin beads to pull them down. So the biotin can be added to the 5' or 3' ends of DNA or to the bases. It doesn't much matter. Okay, well, a very large number of very long probes are used. They're 125 nucleotides long, and they are antisense to the Xist RNA and labeled so that they will bind to it. And they are labeled with biotin. So the DNA will be cross-linked to the RNA in the cells so that the two can be isolated. The cross-linked DNA RNA complexes will be digested to create smaller pieces, and then they will be pulled down by the streptavidin-coated beads. Then the precipitated DNA will be sequenced. Okay, again, by the next generation method. So what the DNA sequences will tell us is where the Xist RNA was located in the cells. Okay, so in most cases, the location of the DNA would be the chromosome location where they're made, but in this instance, the location of the DNA, which is associated with the Xist, will be anywhere along the X chromosome. So the inducible system means that the cells are not expressing Xist, and these are going to be embryonic stem cells, and then we will induce their expression, and then watch the induction of the RNA, and watch the way in which, or the kinetics by which, it spreads along the X chromosome. So the Xist RNA expression was experimentally activated in cultured cells, and then we were looking at, first of all, fish to see where the RNA, when we get the appearance of an RNA cloud, but more importantly, using the RAP technique to identify locations where the RNA is interacting with the DNA. So here's a fish experiment of the cells that early on at 0 time, there's no Xist cloud because the cells are not expressing the Xist RNA. The inducer is added, and after less than 1 hour, a small tiny spot of Xist RNA is seen. After 3 hours, you have a larger cloud, and after 6 hours, you've got a very nice large cloud of Xist RNA, which is coating the X chromosome. Okay, so remember about our models. In model 1, we start off at the Xist locus itself, and then diffuse along the length. In that model, we will get binding at very close sequences first, which will subsequently spread to more distal locations on the X. Okay, in model 2, we have a different idea, in that it somehow is able to hopscotch from the Xist locus itself to distal sites in a very short amount of time. And this is believed to occur by proximity transfer, in that those pieces of DNA are actually brought near enough to the Xist locus itself that the newly synthesized Xist RNA can go to different locations because it's being visited by these different chromosomal locations, amazingly. Okay, so here's what the results look like at 0 time. Of course, we're going to be sequencing the DNA that's brought down by the streptavidin, etc. There's no Xist early. We start the induction, and amazingly, after 1 hour, of course, we got a huge amount at the Xist locus itself, But even after 1 hour, there's already Xist present at the extreme ends and throughout the length of the chromosome. It's not fully fleshed out yet, but it's not just at near the beginning, near the Xist locus itself. It is rapidly spread to many distant locations. And as time goes on, you get more and more Xist throughout the chromosome to get a reasonably uniform label across, or identification really, across the chromosome. So this is not consistent with the simple diffusion model. It more involves some kind of proximity transfer or chromatin looping model to get all those distant sites to get to the Xist RNA at such early times. Okay, so this summarizes what I just said, basically, that the RAP will detect the DNA with which the Xist probes were associated reasonably. And you can see the induction by fish along the 6-hour time course. And you can see as the time goes on, we get more and more of the fish signal from Xist. Okay, so we can look at the DNA sequences to which it's bound, and we conclude that Xist is not spread by a simple diffusion mechanism because it appears very quickly at distant sites and continues to accumulate over time. So the simple diffusion model clearly doesn't work. Something involving the three-dimensional architecture of these chromosomes is needed to explain what is actually seen. Okay, so we're not going to describe the chromosome conformation capture technique. It's called Hi-C. Okay, but what it basically does is identify what DNA sequences are near each other or can contact each other in the genome. So the idea is that DNA sequences that are very far apart from each other on the linear chromosome can actually come into contact through chromatin looping and other mechanisms. And this is a very dynamic process, so the chromosomes can compact and rearrange their loop structures. So we're going to compare the results of early Xist binding sites with locations that the Xist DNA locus actually contacts during the time of the experiment. So this is kind of a hard slide to look at, but the little red dots represent early locations of Xist RNA DNA interaction. So what this is telling us is that the Xist RNA is delivered to fairly distant sites fairly early in the exon activation process. Of course, a lot of it is at the Xist locus itself, which is as expected. But nonetheless, they're very distant sites which have already got Xist on them even after a short amount of time. So what this correlation is, the blue dots, is DNA sequences which are actually seen in contact with the Xist locus by the Hi-C procedure. And you can see that the little blue dots largely overlap, the little blue dot peaks largely overlap with the little red dot peaks, implying that those DNA sequences that got Xist early on were ones that where the physical DNA came close enough to the Xist locus to actually capture an Xist RNA, even at those early times, which is kind of amazing. Okay, this is what I just said. So the blue dots are where the Xist DNA is in close proximity, and they match the red ones pretty well, implying that the site of Xist transcription is brought near enough to those other locations that the Xist could be delivered to those locations, despite the fact that they're actually far away on the same chromosome. Amazing. Okay, so the model for thinking about this is that the Xist DNA is cranking out Xist RNA, and that because this region is being visited by all these other sections of the chromosome, that the RNA can go rapidly and easily to sites which are actually physically distant on the chromosome because of chromosome looping and DNA reorganization. Okay, so overview of nuclear organization and TADs, which are topologically associated domains and compartments. We will spend Monday's class talking about these and what's known about them.